understanding genetics, at least we have some information, we have some ground knowledge that will help us to decide better when we want to conduct animal breeding. Okay, so because animal breeding is very massive thing, so we need to have some clue to help us to decide better, to do to come up with the best decision. So we want to make sure that the breeding strategy is effective. Otherwise, it's just a waste. Okay, so this genetic principle or genetic information, they provide the information in terms of the traits, how the quality of the traits, how the traits are inherited and passed down from the parents to the offspring, and how much the trait will be inherited, 100%, 50%, and etc. So it's, as I mentioned, it will be a waste if we don't have this knowledge. We want, for example, we want to breed the animal for milk quality, for example. We select the animal based on the production only. So we select, for example, we select a female because this female, they produce lots of milk. We select them. So we select this female to become the dam for the next generation. However, if we didn't do the genetic study or we don't get the genetic information of this female, we might miss which trait that really contributes to the high production of milk. So if the trait is not inherited, so the next generation, they won't produce as much milk as the parents. So that's why we need to understand the genetic and how effective is the trait, how much it can be inherited and expressed in the next generation. So basically, we need to start with the Mendelian inheritance. So I think everybody already heard about Mendelian, basic principle of inheritance, including dominant, recessive, alleles, and it explains how traits are passed on. So how do we get this information? For example, if we select some livestock like cattle, so we can start from observing the phenotype and then genotype of the animals, and we need to observe certain generation to be able to understand which is dominant allele, which is recessive allele, and we need to conduct some molecular genetics to be able to get the information. So other than that, quantitative genetics. So this is the branch focus on the trait influenced by multiple genes. So for example, when we talk about milk yield, it's not only one trait that contribute to the production of milk, how much milk is produced. Not only a milk production trait, it's also involved in terms of feed conversion ratio. For example, feed conversion ratio trait will also impact on the milk production. Other than that, probably body weight trait, or the growth of the cattle trait will also contribute to the production of milk. And other than that, probably the size of udder, okay, it's also will contribute to milk yield. So this one trait, this one aim is not based on a single trait. It's also involved in multiple genes that contribute to the same outcome. So we need to understand how they are interlinked together. And then, of course, by the aid of molecular genetics, modern technique like DNA sequence, marker-assisted selection, this will provide us some information, will provide the insight into genetic makeup. So from this, we can compare all the information, and we need to map on which gene relate to this and that genes, and how much that gene affect this trait, for example, and how much this trait can be passed to the next generation. So basically, it's a huge puzzle. It's like a huge, excuse me. Excuse me, someone is talking. Okay. All right, so basically it's like a huge puzzle with the pieces is everywhere. We need to sort all the pieces in place to be able to come with the best effective breeding strategy. Okay. Right, so this is the main source of the data for animal. So when we talk about breeding, we need to understand genetics. We need to understand what are the principles for genetic information, including the phenotype. Phenotype is combination between genotype of the particular animals and the environment that they are raised on. So that is the phenotype. And for genotype, genotype is the genes information within the animal. So combination, phenotype and genomic data will help in terms of improving faster genetic improvement. All right, so here are the examples of the genetic factors that cause genetic and environmental factors that cause different effect to the animal. So genetic factor like gene selections, genomics selection. So by knowing this, by having this information, we can combine with the other information in terms of phenotypic or the performance of the animal, in terms of their observation, for example, age, impact on the reproductive performance, the conception rate. So how is, for example, this one sheep, we get all the information related, not only genetics, the physiology, the other reproductive information. So we combine all the information and we can conclude how good is this animal? How good is this animal actually? And how good is this trait can be passed over to the next generation. So for basic principle of animal breeding, the first thing we want to do before we can conduct animal breeding, we need to do selection. Okay, so selection is very important. So that's why before we want to select, before we want to decide which animal we want to select, we need to understand the genetic background of that particular animal, genetic background and the physiology background of the animal. Then only we can do better selection. So if we have, for example, if we have a farm of sheep, let's say, so if we want to select the female to be the mother for the next generations, so if we compare, if we select the female based on the physiology only, we might select the biggest one or the prettiest one and etc. Or the first one that reached puberty and etc. So, but if we have additional information in terms of genetic information, so we can combine both information, we can have better conclusion on the animal before we can do the selection, so that it will improve the accuracy of the selection. So we need to choose the best animal based on the desirable traits like health and productivity. It depends on what trait we want to produce. Not only that, we also need to combine with the genetic information to increase the accuracy. So the more information we have during the selection, the better selection we can do. All right, so that is the first principle. And then after we have decided which male or which female that we want to use or we want to maintain as a breeder, so we need to decide what kind of mating system or mating strategy we want to apply. So it's either we want to do inbreeding, outbreeding, or crossbreeding. So all these mating system or mating strategy, they have different, they will produce different outcomes. So to achieve optimal genetic outcomes, we need to decide which one is the best. For example, go back to the main objective or the main aims of as a producer. Of course, let's say when we talk about the sheep herd just now, as a producer, if I'm the producer of the sheep herd, of course I want to make sure I choose the parent stock that can produce lots of offspring and can produce lots of milk as well, to make sure that the offspring can be sustained. And then also in terms of size, I want them to have a better weight, good body weight. So for that reasons, so I need to decide whether I want to go for inbreeding, outbreeding, or crossbreeding. Let's say in my farm, let's say I'm in Kelantan or I'm in Malaysia, I have one herd of sheep that is the best in all other places in Malaysia. So do I want to do inbreeding? Do I want to do outbreeding, or I want to do crossbreeding? Since I already have the best sheep in the whole Malaysia, none of other sheep as good as my sheep. So of course, I want to make sure that I can sustain the best sheep in Malaysia. So if I do outbreeding, I might reduce the performance of my sheep because I already have the best sheep in the whole Malaysia. So I need to decide whether I want to outbreed or not. So if no other good sheep in Malaysia, of course, I don't want to do outbreeding. So no outbreeding. So probably crossbreeding can be done because I already have the best doppel sheep, okay, one breed of sheep, doppel sheep. So I want to improve, I need to try to get another breed of sheep, but the best performance of the sheep, that particular breed. So I can do crossbreeding. So that's why back to the objective or aim. So we need to decide what kind of mating system we want to conduct, or producer want to conduct based on the objective of the breeding. But then let's say if I am the geneticist, so I want to, or I'm working at the genetic preservation center or something like that. Okay, so of course, if I'm the worker there, the objective of bank gene, of course, is to sustain the certain particular gene, certain particular purebred. Let's say doppel sheep, pure doppel sheep. So for that reason, I need to do to choose wisely what kind of breeding system to make sure that I can maintain the pure breed. Okay, so again, back to the objective of the breeding. So what is the main thing we want to produce? Is it to improve the production? Is it to maintain the genetics? Is it to expand or evolve that particular breed, or etc.? So basically, the mating system, there are different kind of mating system for different objective. And then next, we need to understand heritability. We need to understand how much of a trait we want to produce can be inherited into next generation. So we need to understand how much of the traits is determined by genetics versus the environment factor. We need to include as well environmental factor. And then we can come up with a conclusion to decide which traits or which animal that have a good heritability on certain traits that we want. And then, so for heritability, we need to have like different series of or different generation of the animals for us to be able to understand the heritability properly. And then next, genetic variation. We need to ensure that there's enough diversity of the population. It's very important to avoid the genetic bottleneck. So we can maintain the resilience against the disease, environmental changes, and etc. So for genetic variation, for example, let's say for doppel in Kelantan, probably normally they will have different performance compared to doppel in Johor, let's say, or doppel in Sabah, Sarawak, or doppel in Indonesia, or doppel in other country because of the environmental factor. The animals or the genetics of the genes inside the body, it will evolve. It will evolve based on the environmental factor. All right, so for selection methods and techniques, we can conduct either mass selection, which is we choose best of the best based on the performance for the particular traits that we want. And then other than that, pedigree selection. So for pedigree selection, we need to have information of the ancestor or the family of that particular animal. So we need to analyze, we need to have the information of their family history to identify what are the desirable traits that can be passed down to the next generation. And then next is the progeny testing. So progeny testing are also important to evaluate the performance of the next generation, the offspring, to assess the breeding value of their parents. And then following that, we need to do performance testing to measure the individual's animal for specific trait. For example, the production, growth rate, and etc. So the more selection technique that we conduct, the better outcome will be. It will increase the accuracy. So when we talk about the selection, this is the phase where we don't conduct anything on the animal yet, but we study the animal first. We study everything about that particular animals before we want to select and decide. So selective breeding techniques involve phenotypic selection, which is identify the animal based on their desirable characteristic, and then evaluates the pedigree, the parents, the ancestor of that particular animal, and genetic evaluation is also important to assess the underlying genetic potential of the animals using molecular technique. All right, so breeding programs for improved productivity. So by developing effective breeding program require careful planning and implementation to achieve long-term success. Okay, so not only genetic information is important, selection and mating system also important, but another important aspect that sometimes people forget to practice is record keeping. So it seems very simple and some people say not very important, but actually record keeping is very important because this is the other kind of information that will be very useful in livestock production. So by record keeping, we can maintain the accurate records of the performance of animal, their pedigree, their health, the conditions, so that we can integrate this observation record with the genetic evaluation, and then we can increase the accuracy of the selection and mating strategy. So next, genetic improvement, so that only we can achieve genetic improvement. So continuously monitoring and evaluate the animal breeding program. Okay, all right. So one example for animal breeding system is crossbreeding or hybrid vigor. So this is one of the best strategy to increase the production or to achieve hybrid vigor. So basically, crossbreeding is combining desirable traits from different breeds, but the integration will produce the beneficial characteristics. For example, meat cattle, lots of meat cattle have been crossbred to increase the performance. So for example, in Malaysia, we have local breed which is Kedah Kelantan. The name of the local breed cattle, Kedah Kelantan. So this Kedah Kelantan, the characteristic, they are very prolific. They have high disease resistance. They are very survivor and etc. But in contrast of that traits, they have a very small body structure. They have a very their weight is not very heavy, so they are quite small in terms of size. So the thing is not everything is bad about this Kedah Kelantan, because even though they are small, but they are very survival. their survivability rate is very high, and they are very prolific. So they can, even in a very critical environment, they can produce offspring. They can produce the calf. But then when we look at the problem, they have a very small size body. So how we can improve that? So the crossbreeding is the answer to improve the production of Kedah Kelantan. So that's why some researcher and government sectors have conduct the research on crossing the Kedah Kelantan. So they keep crossbreed Kedah Kelantan with the other important breed. So to aim for two things. One thing is survivability. Second important things that want to be achieved is increase in production. So by crossbreeding, not only we can maintain a good trait, but we can also import other good trait from other breed into that particular breed. So that is the beauty of crossbreeding. So it will help us to achieve hybrid vigor or heterosis. Hybrid vigor or heterosis is the observation of the performance of the next generation or the offspring of the parents. So normally the offspring will have better performance, will achieve superior performance compared to their parents. That is when we achieve the hybrid vigor. So other than that, increase the genetic diversity. So crossbreeding programs help to maintain genetic diversity when within the livestock population. Okay, when we talk about animal breeding, mating strategy, and etc., for example, if you want to conduct crossbreeding, how do we conduct crossbreeding? Let's say I have Dorper sheep or I have Kedah Kelantan in my farm, and I want to cross to Limousin, for example. So do I have to import the whole animals, or there are other alternatives? So recently, of course, we have other alternative to do that. So we don't have to worry, the producer don't really have to worry that they need to import different kind of breed or anything, or that they don't have to worry if the animals survive or not in our condition, our environment. So with the technology, we can combine with the reproductive technology in animal breeding to achieve the animal breeding goal. So I believe that you already you have already learned about the reproductive technology from previous topic. So I just will summarize the reproductive technology involved that we can utilize to improve the efficiency of animal breeding. So including artificial insemination. So this diagram illustrates how the artificial insemination. So let's say if I want to use, I have Kedah Kelantan just now. I want to crossbred with Limousin, so I can just get the Limousin sperm that have already been analyzed the quality, and then preserve in cryopreservation. Let's say if the Limousin is from Europe, so it must be transferred to Malaysia via flight or ship. So it must be preserved properly before it reach my cattle. So, and then normally the technology used to preserve the sperm or ovum is cryopreservation. And after it reach my farm, let's say I can do the artificial insemination by deposit the sperms into my female Kedah Kelantan, for example, so that I am able to import the breed. I'm able to crossbreed the two different breeds of animals by reducing the cost and time. So we don't really have to buy that particular animal, import here, take care of the animal, and etc. etc. So we can skip lots of work. By technology, we can skip lots of work, and it will help in achieving animal breeding objective faster. Other than that, embryo transfer. So embryo transfer is the process of collection of fertilized embryo from the donor female implant in the surrogate female. So let's say I have a Kedah Kelantan females that are not very good, not producing good ovum. So either I want to cull the animal or I can use it as a surrogate females. So embryo transfer, probably we can also get the good and high quality of embryo from any other country. Okay, so how is the embryo transfer? Is by selecting one female donor, collect the oocyte from the donor, and the oocyte will be fertilized and cultured until its growth up to 5 to 7 days, and then next embryo will be transferred into recipient female. So let's say that donor female, if that donor female is very good in terms of production, we can replicate the offspring of that female that carry the trait which is good in production, and we can replicate in higher number, because when we talk about cattle, it's very critical, because cattle take longer period to grow, to the gestation period, and to reach puberty, and etc. So the growing phase for cattle is much longer. So it's a bit difficult if you want to wait for one female to produce her next one offspring, and then we need to wait another 3 to 4 years for the next generation, and etc. But by implementation of the assisted reproductive technology like embryo transfer and artificial insemination, we can replicate higher number of the production of good next generation. So basically, we can reach the goal of animal breeding faster, sooner compared to natural production. Right. So other option is in vitro fertilization. So basically, it's the process before embryo transfer. So the embryo either inseminated in the donor female, or it can be inseminated in the lab. So if the embryo is inseminated in the lab, we call it as in vitro fertilization. So the collection of eggs from the donor, and then fertilized with the sperms in the culture for 7 days before transferred to the recipient female. So that is the in vitro fertilization. So this diagram summarizes the process of in vitro fertilization. So donor female will be induced by injecting hormones, and then they will superovulate. So they produce many number of oocyte, and oocyte is collected and fertilized in the lab, and then culture and transferred to the recipient female. Right. Other techniques that can be done is cloning. So cloning is the process by creating the genetically identical copies of animal through somatic cell nuclear transfer, SCNT. So this example, the production of the cloning is Dolly sheep. I think everybody already heard the story about Dolly. So basically, the egg cell donor is collected, but then the nucleus from the egg cell is removed, and the nucleus from donor is collected from the mammary gland cells. So the nucleus from mammary gland cell is infused into egg cell of other female donor, and then culture and transfer to the foster or recipient female. And after 6 months, the lamb give birth. The ewe give birth. The foster mother give birth to the cloned lamb, which is the one that provide the nucleus into the egg cell. All right. Others technology is the genomic selection. So genomic selection by using DNA markers to predict the genetic potential of animal for specific trait. So this will guide in better breeding, animal breeding decisions. So it will help in terms of accelerates the genetic improvement, because we only select the animal with the best genetic potential at very early age before we can observe the characteristic of the animals. And then it helps in terms of increase the accuracy of breeding program by focus on specific genes that we want to use for certain traits that we want to produce. So it will also help in terms of reducing the generation interval, leading to faster genetic progress. So this is the process of gene editing. So ovum is picked up and in vitro fertilization is conducted, and then the zygote genes editing can be conducted by edit the gene. So we can change, we can do some amendment to the gene, and then embryo is culture, and then blastocyst will be blastocyst biopsy, and then sequence biopsy, and transfer to the recipient females. And after 9 months for cattle, after 9 months, the mother will give birth to the embryo that have been improved their genetic, have been edit their genetic, so that the offspring have genetic superior genetic compared to the parents. All right, other strategy or technologies is sex-sorted semen. So sex-sorted semen is technique that separates sperm based on the sex of chromosome that they carry. So this also will help in terms of the selection. Let's say if some farm, they only require female. For example, a dairy farm, they want to produce lots of female compared to male. So that this sex-sorted semen technology will be very helpful. So it will help in terms of enable the production of offspring for certain desirable sex that required for the aim of the producer. So optimize the herd composition. It will also helps in terms of increase the efficiency in breeding programs. And then reduce the number of unwanted or less economically valuable animals. So for example, in dairy farms, sometimes previously they used to practice to cull the male animal, because they don't require lots of male, right? So it will cost the producer lots of money if they want to raise the male animal. So normally they just cull the male calf to reduce the cost, because the male calf is not required in huge number in dairy farm. So that's why by using this technology, we can reduce the number of unwanted animals, so we can reduce some unnecessary procedure. Okay, next, the pedigree analysis and herd management. So pedigree record is very important. So we need to maintain detailed records about the ancestor and how the ancestor perform. And then again, genetic evaluation to assess the genetic merit of the individual within the herd. And then only we can do mating decision. Then only we can strategically pairing the animal, so to improve the expression of desirable traits. Okay. All right. So for example, genetic markers and trait selection, this will help in terms of improving the growth rate, select the improved growth and feed efficiency, And other traits that probably important in ruminant production, like milk yield, we can identify animals with the potential for high production, disease resistant, as I mentioned earlier. So we can breed the animals to enhance the immunity, not only for production, but also enhance their immunity. Okay, right. So, however, when we talk about animal breeding, of course, there are some challenges and consideration, some things that we need to consider before we conduct animal breeding. So one of it is the genetic bottleneck, which is we need to maintain genetic diversity to avoid inbreeding depression. So we need to make sure that's why where the record is very important. So we need to make sure that genetic diversity is maintained. We want to avoid inbreeding. So regulation and ethics, we need to ensure that the responsible and ethical practice is used in conducting the livestock breeding. And other than that, environmental factor is one of the major factor we need to consider as well, the influence of the environment, because the environment will cause the traits to be evolved, or to make sure that the animal can survive the environments, or environment change the condition. Okay, future trends and innovation in livestock breeding. So including genomic selection, as I mentioned earlier, gene editing. Now, recently, everybody talk about the precision farming. So basically, it's integrating of data-driven technologies to optimize breeding and management decision. And of course, on top of everything, sustainable is the most important aspect. So we need to make sure that developing breeding programs can prioritize a very sustainable breeds. So by integrating all of the technology with the precision farming, we can improve the efficiency of animal breeding program. So we can start with optimize the reproduction outcome, which is targeted breeding programs. This will improve in accelerating genetic improvement, increase reproductive efficiency. Technologies tool that can be used includes use of different kind of sensors. So the sensor will collect lots of information, data. So all the data will be analyzed, and data analytics can be used to translate all the information. And other than that, including the automation in reproductive management. So for example, robotic milking machine. That robotic milking machine can not only harvest the milk, but also can record all the information of the female that they're milking, their milk. So for example, how much milk, how good is milk quality, how is the body weight of the female, how much she eats, and etc. etc. Lots of information or lots of data is it's required, and it will be very helpful in term by using this technology. It will be very easy for us to translate all the data into valuable information. So it not only helps in terms of increase the production, but also uh, it will cause the impact on sustainability by resources efficiency, reduce the environmental effect, and enhance the animal welfare. Okay. All right. So that's the end. I stop here. So I think I open for the question and answer session.
All right, thank you very much, Professor Dr. Khairiyah, for your excellent and really informative talk. And there are many insight and knowledge that we can pick from your presentation. Maybe we can move to the next presentation. I mean, we will have a the answer the discussion session in the end of the second lecture. All right.
Okay.
I would like to invite Professor Han Jianlin. Hello, Professor Han Jianlin.
Yes.
All right. Okay. Thank you for your time. And now you can share your screen.
Yes.
And
Let
yep.
me go to share.
All right. Okay. You have 45 minutes to deliver your lecture, and there will be a discussion session at the end. And now you can start, and the time is yours. All right. Thank you.
Okay, great. It's really my pleasure for being invited to deliver a short presentation here under the program, what we call the Applied Technology for Tropical Indigenous Livestock. As part of the Hasanuddin Animal Science summer course for this year 2024, it has been quite some time, since I left Makassar, left Burgo, Indonesia, more or less 2 years ago. Now I'm back to work in China. As the principal scientist in charge of the program of the small ruminant, in particular the domesticated sheep and goats, at the a new facility the country built last year, what we call the Yazhouwan National Lab, which is based in Khairiyah, in the which is the southmost island of the mainland China. So in a physical position, we are rather close to each other, So today I would talk to you regarding what's really the importance of the livestock genetic resources in relationship to the climate change. I'm sure say yes. People around, online or for this lecture, you are all familiar, more or less, say what we're talking about the livestock, when which refer to the domesticated animals, the poultry species. That means the mammals, the birds, which were domesticated and start probably something like 10,000 years ago till very recent, maybe only a few thousand years ago for some species. While, given my focus at the moment on the sheep and the goats, they were domesticated around 10,000 years ago in the Fertile Crescent region in the Near East. After the domestication, they were migrated together with the human movement, because we are talking about the domesticated animals. They are basically very different from wildlife. For wildlife, they are free ranging, they move around themselves. But for the domesticated animals, they are under the full control of humans for production, to produce meat, milk, egg for our daily life. So in terms of the major species, we are talking about cattle, chicken, pigs, sheep, goat. Among say, okay, we have also other minor species like buffaloes, like horses, like ducks around, as you know. I will mention them in the later slide. When we are talking about domestic animals. These are the commodities support what we talking about the livestock production. When we talk about the livestock production, say I think Indonesia, similar to a country like China, where the livestock production may contribute to 1/3 of our total agricultural production value. Say they contribute 1/3, to the agriculture sector. While we look at to say, yes, how this 1/3 of the proportion was derived, because I think we calculate the raw value, say what milk at the door of farm, what the price of liter milk or a kilo of meat for people to come to buy from a farm. But we know for livestock product, which is different from other, say, agricultural produces like rice, like wheat, a livestock product of meat, milk can be processed, packed, and marketed through a very long, what we call value chain. So along this chain, the value of the milk or meat is added at every step. So from the farm to the table, we buy the product back home for consumption. So the meat was slaughtered, was processed, was transferred, and similarly for egg, for meat, for milk. So the value from farm to our meal table, so the meat, the price increase maybe 5-10 times. For milk, similar. For egg, maybe a little bit more. But for rice and maize, you can think, say, okay, we buy rice from market, but rice, they are not processed, not packed, so complicated compared to meat or milk, because to pack milk, to market meat, you need to follow a high level or standard of quarantine protocol to make sure the meat, the milk quality are maintained during the marketing, during the value chain. But rice is simple, maize is the same, very simple. As long as they're dried enough, you can take them whenever you go, you can sell them directly. You don't need to really to pasteurize them or to pack them. Although we do it, we see in the supermarket, the maize or rice, they are salted, they are labeled at different price, but they are much simple compared to meat and milk. In this case, you can see in this slide here, what we are talking about, the value, what we call a commercial value of the major commodity from agriculture. So these are the value we pay for, as a consumer to buy them for our. See among the top 5, the agricultural commodities, cow milk, rice stay at second position. Cow milk stay at first, which is the highest value we paid already. Then the third is the pig meat. The number 4 will be the cattle meat. The number 5 is the chicken meat, followed by fish meat. So I mentioned, say, the livestock production contribute to 1/3, but if you look at the commercial value at this level, livestock is really much more important in terms of the contribution as a commodity to our daily life. We pay much more money to meat, to milk, to egg than we pay for rice. So in this case, apparently livestock is much, much more important than let's say, rice production or maize production. Given, say, a country like Indonesia, we eat much more rice, but we know, say, yes, goat meat, cattle meat, and chicken meat, they're consumed at quite a lot. We pay a lot of money for that. They are much more expensive than rice. So this is where you can go to argue with your senior, with your management, in the government to say, why don't we get enough share of the budget for research on livestock compared to, I'm sure, say, in Indonesia, I think many people know, say, yes, government will invest much more money in the for research on rice, not for cattle, because in the government side, they believe the rice is more important, is the staple food to everybody. But if you look at the commercial value, livestock is more important than rice. So this is the way how you can argue what we are doing is much more important. We should get more attention, Otherwise, say yes, back to the issue of livestock production linking to climate change. From the map, you can see this is a sort of map my colleagues at the International Livestock Research Institute prepared 20 years ago, how the domesticated species are distributed across the world. Apparently, say yes, if you look at the general map, we have a major livestock production systems from what we call the rangeland-based. That's where people take the animal to grazing on the rangeland, on the pastures. I see in Indonesia, you have the lowland, you have also the high mountain in the central part of the Yazhouwan, where people do keep cattle, sheep, goat grazing freely. Otherwise, I think in the country, we have much more integrated, mixed farming system where in the cropland, people grow rice. Sometimes along the rice field, we may keep goat around. This is what we refer, say, for most of the farmers in the area, they are doing rice farming, but sometimes they also keep cattle, chicken, goat with them in the household, so they can take the animal to grazing a little bit along the paddy rice field. Otherwise, they can use rice straw or maize stop over to feed livestock. So that's what we refer a mixed system. In the mixed system, depending on, say, the rainfall from south to the north, and also largely depending on the temperature. Different species are distributed in different part of the world. They have quite a stratified distribution for major livestock species. You can see on the right corner, we can see this is how domesticated cattle include the Tauran, the Indis cattle, and also the Bali cattle in Indonesia. You can see the cattle are distributed quite a lot in southern part of the world. South American, like Brazil, Argentina, are the major producers for beef, and also some to southern part of Africa and South Asia, Southeast Asia, where, say yes, in Indonesia, we have quite a lot of Bali cattle around Java Island, but they are less densely distributed in Kalimantan and in Sumatra. So we can look at how sheep are distributed. You can see there also sheep quite similar to cattle. They share much large sort of the geography as because they are both large ruminants. They rely on similar as the rangeland or the feed resources. But for the corner, the right bottom in the corner for poultry, we can see poultry are densely distributed in Europe and in Southeast Asia, while in Africa they are less important. There are not many of the poultry species for Africa. The reason is in Central Europe or in Southeast Asia, where we have this modern poultry farming, like factory-based chicken and also duck production, where we keep the chickens in a factory. We maintain them and let them produce as a sort of machinery system. We can keep many millions of chicken in a small unit of land or a particular house. That's where we can see in Central Europe and Southeast Asia, poultry are more popular and definitely are more important because chicken meat production has the highest sort of ratio of 1 kilo meat to maybe today the highest rate could be 1 kilo meat. You only need to feed 1.5 kilo feed to a broiler breed. So the feed conversion rate is very high for chicken. Therefore, we have a modern factory farming system for commercial chickens in Europe and in Southeast Asia, where the human population is much more dense, so where we need to have a modern system to produce enough meat in a limited land and in a short period of time. And this, that map was drawn in 2000, around 2020 years ago. And later on, my colleagues at ILRI, they continued to make update of the major species. So these maps were produced around 10,000 years ago. It looked to me some species, they have a little bit a shift of their distribution range. Why that shift? First definite is the improvement of the production system, as what I mentioned. Example of poultry production. Say earlier we have small holder farming for poultry in the backyard. One family, we may keep 5, 10, or 20 birds in the backyard. But today, that system is more or less diminished. I think if we go to a village today, in most of the country in the world, it's very difficult to find chickens in the village. And the most majority of chickens are kept and managed in a factory system. So that means to say, yes, there is a shift of production system. Otherwise, for cattle, for sheep, goat, the system or the distribution range is shifted because of the climate change. Some part of the world, is getting less or less rainfall, or are suffering with the increased temperature. Then, the livestock, say particular for cattle, sheep, goat, which are mostly rely on the rangeland system for grazing. So if we have a high temperature, if we have less rainfall, then the natural feeding resource or the natural grass are becoming less and less in a particular area where people cannot continue the cattle or sheep farming in that area. So the distribution range, these systems are shifted under the pressure of climate change. We have another recent update, more or less 5 years ago, by my colleagues again, linking to the major species of chicken, duck, sheep, goat. We can see, say, these species, their distribution are more say centralized in the different part of the world, not like the first map we see for sheep goat. They are some have evenly distributed across Africa, across Southeast Asia, but today we can see some corner of the world where the animal population is increased, while the other area definitely become a sort of what we could call a desert. I think this is maybe some people could understand, say, if we continue to go for the deforestry in Kilimantan, then definitely to say, yeah, the natural grass, this feeding resource will be reduced, then the will continue to be reduced. That's what we are expecting at, under the sort of shiftment of the human life, the change of the human society. When you decide to move your capital from Jakarta to other place, definitely, which will bring a significant change and pressure to a new place in Kalimantan, for example. So back again, when we talk about the biodiversity of farm animal, that is the livestock and the poultry species. We in the world we know there are probably more than 1,000 wild mammal species with a large body size around 5 or 10 kilo, but incidentally, and humans, our ancestors, they selected only, say, very few mammal species. Similarly, also even much fewer bird species for domestication. So today, we are still research on the questions of why these species like cattle, sheep, goat, pig, chicken, horse, donkey, buffalo, goya, bittern, and quail, duck, geese, etc. Why these a few species were selected and successfully domesticated by our ancestors in different parts of the world. This is still a scientific question to be answered. We do not fully understand why a species can be domesticated from the wild form into a domestic form. We don't know, that at this moment. But say, given the rapid sort of the achievement in the genetic, in the genomic research, I think the research, the scientists are getting much better, improved understanding. Say there are some genes. Some genes in the genome of a species is contributing to the domestication, what we call a domestication gene, like a gene control milk production or meat production. There are genes controlling the domestication capacity or the possibility of a species. We working on that. I will give you a few examples later. So we basically working on the livestock production, and we are dealing with very few species. I mentioned cattle, sheep, goat, and down to chicken and duck. And at the international level, and the food and the FAO, the Food and Agriculture Organization of the United Nations, that is an intergovernment organization in the world, supported by all our countries, including Indonesia there. So the FAO is very much more concerned with the issue of livestock genetic resources. Here we are talking about genetic resources at a broad sense. We are talking about all the breed of cattle or chicken, sheep, goat. A breed of livestock is, say, follow the definition for breeding, for genetics on livestock. We are talking about a particular population which carry a unique gene pool. They are not shared by another population. They may have a unique phenotype. Morphologically, we can see them showing something different from another population. That's for agriculture, people defined a term, say breed, as to characterize these livestock or poultry populations. So that's what we talk about, the genetic resources today. So you can see at a global level, for cattle, for chicken, for sheep, these three species each may have around more than 1,000 different breeds across the world, followed by horse, goat, pig, duck, which they have something, a few hundred of breed. So in total, say, around 5 years ago, FAO came up with a sort of a summary of the number of the breed, include the local breed, the commercial breed we manage. Altogether, they are around 8,000 breed in the world for livestock and poultry species. And say, we see follow this rapid shift of the production system. What would happen? Say, when we have backyard poultry system in the village, every farmer, every household may keep a few birds, and across the village, across the island, say we may have many different breeds for chicken. But when we move into a factory-based production system, and which are supported by very few commercial breed, like white leghorn for egg production, that can maybe contribute 90% of the genetic resource for egg layer, while say the what we call the the other broiler breed, they are only 5-10 broiler breed across the world. They are supporting this massive factory production. So along that process, we can see the indigenous or the local breed are disappearing because they lose their importance, and no people is going to keep them. So then the village chickens disappeared, then associated, in that case, many local breed also disappeared. So we know, say, for to develop any breed, maybe people, our ancestors spend a few hundred years or even a few thousand years of time to develop that breed, but they got disappeared in very short time of a few years. This is definitely is unfortunate because this factory-based production maybe is not sustainable because they rely on, this intensive farming, intensive housing condition. Some place maybe the smallholder farmer are not able to afford that cost. Or we can say yes, you keep a commercial chicken, you have to feed them with commercial feed at high cost. Many people cannot afford. I'd say today, under this climate change scenario, we can see maybe the yield of crop is reducing in many parts of the world. Therefore, the feed or food pricing will be definitely increased. In this case, maybe some people will come back to live in the rural to restart this, the village or the backyard system again. And we know clearly, say, yes, this commercial breed, the commercial layer, the commercial broilers, they cannot survive under the backyard or the scavenging system. So therefore, we have to maintain our local breed for the future. We don't know what will happen in the near future if we do not find a solution to stop environment pollution or to slow down the release of carbon into the air. Maybe the climate change will be speed up, then that change will be quickly happen. Then this commercial system will be collapsed. All our next generations, they will have to come back in the rural. And if we do not maintain this indigenous breed for future, would our young generation will be at risk for their life. So I will talk a little bit, say what happened with domestication. Here you can see in the world we have nearly 1/3 of the mammals with the body weight above 45 kilo, and only 10 out of 10,000 bird species they were domesticated. Say, okay, around 10, 15 mammals were domesticated. So I mentioned earlier, say, we are still doing research to understand why these few species were selected, were domesticated. We have no clear answer today, but we know already have many publications to reporting several domestication genes for different species. A process domestication involves, say, a sort of initial association with wild stock. People, our ancestors, they have to find a way to get in close contact with, say, with a baton, a wild bovine species in Indonesia. Before you domesticate the baton, you have to find a way to get close to contact with the animal, and so that you can, say, find a way later to control the animal. Say you build a shelter for them so that they can come close to your house, stay around with your house. That's where, say, the first step. And say at later on we may decide to say, okay, they can come regularly to the house for food, for water. They are establishing a sort of contact with humans, with us. We may control the reproduction, and we may keep the young or the newborns of a baton, a calf of a baton in a shelter or within the captivity for breeding. If we can get success of a captive breeding, which means to say, okay, we are close to the full domestication of that species. That's the first step. After domestication, more or less say we are able to tame, we can control the reproduction in captivity. Then we may start to make a plan how to breed them, how to improve them. We can say, okay, a baton may produce a calf maybe in April every year. Then under the human control, and we may control the natural mating by separate the male from the female. Say, we may manipulate the calving season in other ones. Say maybe they can giving a calf, a baby, a baton or baby body cattle after domestication every month of a year. In this case, we are controlling the breeding, we are controlling the reproduction so that we can get a regular supply of meat from body cattle. And say at near say modern days, people are not satisfied with the slow growth rate of body cattle, and they may start to cross body cattle with other cattle breed to make a new improved breed. That's what we talk about, the breed improvement. So say yes. If you look at based on the archaeological evidence, we have what we call the agricultural revolution, which took place around 12 to 14,000 years ago. This is a revolution, say, only after we managed to domesticate animal and plant into a livestock or crop under the full control of our hands for a planned reproduction, for planned breeding, and the human society will be able to get enough food at a consistent level of a year. We do not suffer hunger, say in the summertime or in the winter, and because we are not controlling everything of crop and livestock, we can make a plan when we grow them, we plant them, we produce meat or produce rice for our consumption. So this is very important for the expansion of human population, leading to the settle down, leading to the establishment of our modern society. Without, say, the agricultural revolution, we may still struggling today into the jungle to catch a bird, to catch a baton for food, which it is a lifestyle of opportunism. So mean to say today you may be lucky to catch a jungle from in the jungle, but maybe next 5 or next 10 days you are failed to catch anything, then you and your family will be suffer from hunger. So you can imagine how important this agricultural revolution. Although say yes, in the 2 century ago, we have what we call the industry revolution, where people discovered the machine, people discovered the electricity, discovered this mobile equipment, etc. That's what we refer to this most latest to say the revolution of industry. Otherwise, say all those are supported by agriculture first. So during the domestication, we humans, we are able to say to domesticate the species, and that species may evolve, may develop, say, a new morphology, new genetics to meet or to live, to survive under the human control. So here, one of the examples is the body change, body structure change of a wild boar into a domesticated commercial breed today. You can see for wild boar, they have much more body size, 70% in the front part, because they are struggling, they are hunting in the jungle for food themselves. And after the initial domestication, the front part is reduced. Our indigenous pigs in say in Sulawesi, where you can see today the pigs, they have a 50% front and the rear part. But if you go into a commercial system today, the commercial breed, the rear part is taking up 70%, while the front part only has 30% of the body weight, because most of the meat from pigs, and even the same for cattle, most meat are coming from the rear leg, not the front part. So most of meat are here, and we continue to improve the pig, improve the belly cattle into a more productive breed, where the rear part are fully developed with much more muscle. That's what we are talking about meat. So based on this archaeological evidence, we can see across the world, there were only a few, 1, 2, 3, 4, what we call the Neolithic agriculture homeland linking to livestock production. For the first, it was involved something like Turkey. For the second, in South America, where the llamas, the llamas, alpaca were domesticated. And in the Fertile Crescent region, Near East, many species were domesticated 10,000 years ago, including cattle, sheep, goat, even pigs were domesticated there. That's the number 4 in the east part of China, where the lions, where maybe the pigs from East Asia, buffaloes, chickens were domesticated. And we see the climate change is a phenomenon taking place most part of the world, where the weather is scattered, and it's very difficult to predict weather tomorrow based on the data we collected over, say, in the past century. It's very difficult to predict if it is going to rain tomorrow in our area, because the climate change, the pattern is localized. Even say this village rain today, but next village there's no rain, it's dry. So that's what we are talking about, the climate change. The rainfall reduced or increased in a particular area, or at the temperature at the global level is rising up. Then, therefore, I mentioned, say, the first, say, the plant production, the biomass pattern is changing, most likely is reduced across the world. Although maybe a few places on a particular island, maybe the rainfall is increased, the biomass may be increased, but in general, we will suffer from drought. we will suffer from a reduced, say, biomass or reduced capacity of rangeland. On the other hand, say yes, while the climate change is affecting livestock production, where we may have more disease because of the pathogen shift, and we may have limited food resources, and we have limited water to drink in a particular area. We have to take animal over a long distance or to build an irrigation system for drinking water of human and livestock. On the other hand, say the livestock production is also contribute to the climate change. Livestock are releasing the methane, releasing carbon quite significantly. That is a part of the agriculture production. We are speeding up the climate change. While one side we are affected, on the other hand, we are contributing to the climate change. We need to find a solution to slow down, the release of carbon from livestock production. So I think I mentioned already the heat stress will be an issue. Then this, most of the intensified livestock production will be affected because of the limited availability of feed and water resources. So these are all the issues we will be facing. So over my life, we have been doing this characterization of the livestock genetic resources. We want to know where a species was domesticated, why they are distributed across the world today, how they were migrated, what number of breed we have for a particular breed, at a phenotypic level and also at a genetic level, what refers to the genetic diversity. So we know, say yes, today we have modern tools to quantify the genetic diversity at the DNA level. We can examine the DNA polymorphism, the DNA mutation, DNA variation, using genomic tools, including the microarrays, the whole genomic sequencing, are become more, affordable for us to do research. So I will give you an example, say, for buffalo. We do have, say, the Khairiyah buffalo, the spotted buffalo in Indonesia is the most expensive in the world. I visited the market in Toranjan. It was really amazing. So how people, they appreciate the value of a particular buffalo for their ceremony purposes. So that is also give us an impression that the livestock genetic resource is very different from a crop, because for crop is a handful of seed. You do not have a feeling, you do not have really association to a rice seed. But to people, we do have a close contact to a Toranjan buffalo or any other animal we keep. We give a particular name. We love that animal, it's different from rice. So the value of livestock genetic resource is not simply the productivity. They have the cultural importance, they may have a religion, purposes, so it's much more complicated. For buffalo, we have been doing research to understand what's the genetic differentiation of the different breed. In a particular country here, I give you an example, we work on the Iranian buffaloes. We managed to collect different population breed in different parts of Iran. We do the genomic sequencing, we build a phylogeny, we do the principal component analysis to differentiate one population from the other population. At the end, would seem to me say the Iranian buffaloes are, although they have 4 or 5 local breed, but they can be classified into 2 genetic groups. So that means to say, okay, maybe 2 of them or 3 of the population, they share same genetic background because they are distributed nearby. So they have a gene flow between populations. Therefore, they share genetics, they share similar morphology. We should not treat these populations that separate breed. They should be combined into one for genetic improvement, for conservation. While the other population is isolated, they have developed a unique genetic background, which should be pay much more attention for to treat as a different breed. And we did the whole genome scan. We were able to identify genomic region on the chromosome 2. And we see on the chromosome 2 in this area, they have a totally different, say, diversity pattern. So the level of diversity in this region significantly reduced. And we can zoom into the region. We can see the genes within the region. Then we can examine the function of the genes, say, okay, maybe in one population, these genes are more important, which is under significant human selection, where the diversity of that gene is reduced. We can come up with tools, what we call the molecular marker, to support the breeding program. The same thing, we have many different breeds across the world. They have a totally different morphology. If you look at the tails are very different, from very large fat tail into a very small tail with no fat, or there is no tail, but the animal, they involve the capacity to store fat around the rim for a hard time. Say they store fat as part of energy to be mitigated later if they don't have enough growth. They may use the fat stored in the body to survive. And we can collect enough samples looking at breed of sheep live in the high altitude or low altitude. We can sort the genetic or the genomic pattern into 2 groups for low altitude, high altitude sheep. Then we can scan the genome to identify genes associated with the high altitude adaptation or with the drying, the adaptation to dry environment. And by doing that, we can come up, a list of functional genes associated or responsible for adaptation to high altitude, adaptation to dry environment. And so if we know the gene, we will definitely come up with a molecular marker for breeding to speed up the genetic improvement. On the other hand, say yes, we know that we have many bovine species. In Indonesia, we do have Binti, we have Bali cattle. It's a derived form after domestication. Then say yes, we know that the Bali cattle is continuously used or somewhere, say they hybridize with Bali cattle in the rural areas. Say there is a gene flow from the wider population to the domesticated population today. In this case, by this interspecific hybridization, the genes are coming back again from wild into the domestic population or transferred from one species into another species. This process can, say, increase the diversity of a particular breed of cattle. We were able to identify what we call the integration signal in a genome, a particular genomic region, say a cattle breed may carry some alleles of a gene integrated from another species to support their adaptation to a particular environment. That's where this gene or this DNA mutation can be used for breeding to improve a particular breed. So we did the same. We were able to identify the integration of Baton or Bali cattle into the local cattle in Southeast Asia, where the Bali cattle has been, living in the Southeast Asia in Bali or in Java for maybe many millions of years. So they are strongly adapted to the local environment in Southeast Asia, while the Indian cattle, the zebu cattle, were introduced from South Asia into Southeast Asia. They can meet with the Bali cattle. They were able to hybridize the cross so that they acquire some genes from Bali cattle for improved adaptation in a short time. So by doing all this genetic analysis, we come back again to look at this world map. We are now able to identify more than 10 centers of livestock domestication. At the beginning, I told you there were only 3-4 centers of domestication based on of the archaeological evidence. But today, with the last 30 years of research in genetics, and in particular the genomics work done in the last 10, 15 years, we are able to identify many new centers of domestication linking to particular species like Bali cattle in Java. The Bali cattle was domesticated locally in Java, as that's where even we have other Madura cattle or whatever. They are a hybrid origin of the zebu cattle domesticated in South Asia, and they were introduced into Indonesia, and they were crossed with the Bali cattle to make up another new breed. So that's where we can see over the last 10,000 years, many human societies were involved into the domestication of livestock species, and our ancestors all contribute to the agricultural civilization. And at the end, I would say today, if we look at it, the genetic variation, we see the livestock, poultry, the breed. There are two sources. The first is the one, the genetic diversity inherited from the white ancestor directly over the domestication. And the second source will be the integration. The integrative hybridization between different species. By doing that, under the human control, we were able to harvest the genes from wider species, a wider relative. Definitely the two species, they have to be able to cross, and their progeny have to be able to reproduce themselves. There should not be what we call a reproductive isolation. If there is isolation, there will be no gene flow. And only say if there's no isolation in reproduction, then the genes can be transferred from one species to another. In that case, the diversity of a particular breed or species increased as a new source of variation that would contribute to the diversity of the morphology, the productivity, or the adaptation to environment, which is changing every day under this process of climate change. And we are doing a lot of research in the last 10 years with the genomic tools to identify or localize many genes in the genome of particular species or a specific breed, like a Bali cattle. I know many of our Indonesia colleagues are doing research to identify genes associated with a particular trait of Bali cattle. That is where I say yes, with modern achievement, we will be able to identify specific DNA mutation as a molecular marker for breeding. And they say today we have modern tools like DNA chips. You can genotype 10,000, even 100,000 DNA mutation at the same time for one sample at very low cost. In this case, we can really examine the DNA variation across the genome of a breed, which will come up with, say, the base idea how the different breeds were developed over the history. We can say yes, the phenotypic diversity is developed based on the interaction between genes. Sorry, genes with the environment when the animal are kept. You can see that these are the contours of, say, resampling different breed of cattle or horse. We know that there are many different breeds. They look differently, or even they maybe look similarly, but the genetics are differentiated. By doing the research, we can know today to looking at, say, at a village, say, for barley cattle, for example, we may collect 10,000, 5,000 barley cattle samples, and we can do the genotyping using DNA chips, and we can quantify the genetic composition or the genomic composition of individual cattle. Most likely, the barley cattle today they have a genomic background like this. Some animal where maybe they have some stable genetics, while they may maintain the Banting genomics also. And say in a particular production system or the management scheme, we may decide to have a particular proportion of zebu to Banting or to barley cattle genetics. We decide to develop a new population with a fixed genetics in this proportion. That's what say yes, using the genomic tools, we can manipulate, we can give you the accurate estimation of an individual genetic background so that you can quickly develop a population of animals that share the same genetic composition. That is means this population will be called a breed, which have a stabilized genetics. And by following the next generations of the reproduction, the newer progenies will share the same morphology, same productivity, and which will allow us to have an optimized management system, feeding system to increase the efficiency of livestock production. And say based on the genomic research, today we know that we have many methods to deliver our research product. We can come up with a recommendation for new breed. So maybe a local breed is not good enough for that production system or for that area. We know there's another breed to replace, to be used for replacement.
Excuse me, Prof.
Yes,
time is
yes.
2 more minutes.
Yes, I'm finishing. So then we have said, no, we can have a planned crossbreeding program, not like earlier we do, crossbreeding without a clear plan, without a good idea. Today we know their genomic background. We can have a controlled mating system to make a specific different breed. Then say we can do within breed selection to continuously increase the productivity of a particular breed, or at the end, like many advanced countries doing. We have what we call the genome editing tools to create or to transfer or to manipulate the genome mutation at one generation. We can change a nucleotide into another form to make a new function. For example, we can control the Varroa gene of sheep to make the one nucleotide change so that the sheep can produce lamb from single lamb into a twin or triplet per liter. This is what we call the genome editing. This is really the latest tools we are using at the moment, because we can create a unique identical animal in one generation, which is different from other traditional breeding program, which may take 3, 5, 10 generation, 10, 20 years of time to come up with a new population, a new genotype. So at the end, I would say with the rapid development of all the research around genomics, the livestock production is improved. And also, the more importantly, we are trying to match the production system with a breed, to improve the efficiency. Not necessarily we all go for this factory-based commercial production. Even the rural, in the community level, we can improve the livestock to, for better efficiency. So this is we need to have all options for all species. The idea is to optimize the utilization of all the natural resources while to reduce the footprint of livestock production to environment. With this, thank you for your attention.
All right. Thank you very much for your insightful lecture on livestock genetics. It was fascinating to delve into how genetic improvement can significantly impact the productivity and overall livestock quality. All right. Now I open the discussion session, and now there are already 3 questions in the chat box.
Okay.
All right.
See.
For the first question from Rahma, with 2 degrees Celsius increase in environmental temperature, 7 to 10% of livestock are expected to become extinct. The majority of breeders in developing countries are traditional breeders. What will happen to these traditional breeders who heavily rely on natural conditions? Is there a possibility that traditional breeders will become extinct in the future? Please answer.
Yeah, as what I already briefly, mentioned, we are all in the developed country, definitely under much more pressure from the commercial program. I mentioned, for government system, in the government, they think, okay, we can go out to the country, buy cheaper meat, cheaper egg, and to make people happy to live, while they may ignore the local, livestock production, because we are seen as a low producer. The efficiency is not high. Yes, when the temperatures increased, for smallholder system, we cannot control the environment, but for the factory, for poultry, they can make air and conditioned system environment for poultry production. That means to say, yes, there are a way for in the commercial side to growing to continue the business to produce cheaper food. But I think in the developed country, if you look at the smallholder farmer, we have to find a way to live because as what I mentioned says, the livestock is not simply a piece of meat or one egg. You know, farmers who keep animals for many purposes. So we need to monitor the change in the environment. We need to think, say, for follow the rising temperature. We may looking at, say, the heat shock genes or the heat tolerant genes as a molecular marker to improve the local breed for heat stress. So this is things, you know, we have to do. Why in the north for the European countries, they may not concerned about say heat stress, but I think in the south planet, you know, in South China, Southeast Asia, down to Indonesia, I think heat stress is always a problem. As what I mentioned, says the pattern is strongly adapted and body character is good enough. Why don't we continue to work on the body character for improvement, to harvest the genes for adaptation so that we can have a unique production system for body cattle? Why we need to introduce threshing cow, which are suffering in the, you know, the hot, humid environment. So I think there are solutions. We need basically to study more, to understand our, you know, the uniqueness, the importance, and also the specific genetics of our local species and the local breed. Thank you.
All right, thank you, Rahma, untuk your question. And the next question coming from Amrullah. In Indonesia, especially in Madura, East Java, there is a Madrasin cattle breed derived from crossing Madura or local breed and Limousin cattle breeds. The F2 offspring of the cattle have many reports of ovarian hypofunction, which makes a lot of losses, especially in the service per conception of cattle. The case keeps repeating itself. How does molecular genetics respond to this? Is crossbreeding between local and exotic breeds destructive?
This is definitely what we have been talking a moment ago. Say yes, Madura cattle is good enough because they have been developed locally. I think the Madura cattle has a lot of blood from Bali cattle, so they are very strong for adaptation. While the Limousin is really a large-bodied cow. Definitely, if you look at the production, say yes, they can produce a lot of beef per animal. But as what you mentioned already, you notice, say yes, the Limousin cattle, I see most likely they may be suffering from the heat stress, where the reproduction is mainly affected. That's what we know, say the adaptation. The first thing to do is say yes, the animal may slow down the growth, they may stop the reproduction because they are failed in produce quality, sperm quality, eggs for a healthy egg. So following the cross in this case for F2, you know there is an issue for ovary development. Why don't we do the research to see which gene from the Madura cattle is play a role in association with ovary function? In this case, if we identify the gene or the specific mutation from Madura cattle, I'm sure the Madura cattle in genetic side is very different from Limousin. they must carry very unique genes or unique alleles with specific mutation to be functional with our function. Then we, as what I mentioned, we can develop this into a molecular marker to screen this crossbreeding population to select the animals which carry the Madura allele for next generation. We may excluding the animals which carry the bad mutation from the limousine. While say yes, we know which genes, which alleles from limousines contribute the fast improved growth rate. We should use it. That's what to say. If you are working with 10, 100 genes or many mutations, you may come up with low density DNA chips to build all the mutation important from Madura, unique from limousine, make a combination of a set of alleles for a new to screen the crossbreeding animal for next generation. So that's they say yes. I think the genomic research is playing a much more important role along this issue. Thank you.
All right. Thank you, Mr. Amrullah, for your question. And maybe this is the last one. Unfortunately, we don't have a chance to ask our first speaker because she is already left. Okay, this is the last question for you, Professor. Coming from Aulia, and the question is, what do you think about the effect on livestock diversity impact on climate change? Is there any possible DNA change to animal in the future, morphologically? Then you so much, Prof. Thank you so much, Prof. Okay, you can answer.
Yes, this is definitely the interaction of gene along with the environment, while say follows the evolution. Animals are also changing over time, follow the changed environment. So today, say for livestock, we are controlling these particular animals. Therefore, we are able to identify the function of the genes, and we can monitor or we can predict the future climate, the future environment, the future temperature. The next 20 or 50 years, I definitely say that environment temperature is going to increase 2 degrees or 4 degrees. That is very significant. The biomass will be changed. Then say yes, we need animal which carry the heat stress, heat tolerance genes to survive in the new environment. Or we may to have an animal which they can have improved water use efficiency. They are drinking less water in a dry environment. So I'm sure, say, by doing the molecular study, we will be able to know all the aspect of the genes, the genome of cattle in this case, or chickens in particular, and we will know the function of the specific gene. And today, even we are working on a new program. We are thinking that we can come up with a smart breed. The breed, they carry a unique genetic system. They can change the gene expression pattern, follow the temperature fluctuation. So if temperature increase tomorrow by 1 degree, one particular gene is expressed 10% more to adapt to that new condition, which means to say, yeah, the breed will be smart because we follow the artificial intelligence, all this new development, we can combine the knowledge from genetics, from genes, from environment changing, make a breed to be very smart. I think for crop people, they're doing something like this. They are creating new rice breed. This breed can change the particular gene expression under specific condition in a smart way. They can, like what humans say, if we feel cold, we put on one more clothes. If we feel hot, we may remove a clothes. We may go for one more shower. So the breed of crop or livestock can be also smart like that if in the near future. We are working on, we are learning from crop scientists. I'm sure the livestock even say, even though, the genome of livestock is much more complicated than rice, which have a much smaller genome size, while the cattle, sheep, goat, they do have a triple the sized a genome. It's much more complicated. But we are learning from our crop colleagues. We hope to develop a new smart breed in the near future. Thank you.
All right, thank you very much, Professor Jianlin, for your explanation. It was really, really comprehensive, and I'm sure that the audience has gained valuable knowledge that can be applied to their work in the livestock sector. All right, now we have reached the end of the session. But before we close the session officially, I will present a certificate for you.
Oh, thank you. Thank you.
Yes, I will display in the screen.
Okay.
Yeah.
Thank you.
All right. Okay, can you see this certificate? All right, I think I would like to invite Bapak Dr. Muhammad Ihsan Dagong to say something to Professor Jianlin. All right.
Okay, I can
Thank you.
see it now. Thank you.
Thank you,
It look nice.
Mr. Farouk. Yeah, Prof Han,
Yes.
thank you very much, Prof Han, for your time to deliver a talk, very valuable talk for this afternoon. And we hope we can collaborate for another research again. And we hope you will come again to Makassar.
Yes, I'm looking forward to come back or invite you, Isan, to my new lab. We are not far from, we are all on near the ocean. So you should make a plan for a visit. We can work on the ship.
I will plan next year, Prof.
Yes.
I will
Thank
plan next year going to your lab.
Thank you very much.
And,
yes, thank you.
We have already several young lecture.
Yes, I can see you have a very intensive.
We hope that the young lecture will maybe they can pursue their further study in your lab.
Yes, why not? Let's renew our collaboration. We should working together more. As what I mentioned, I'm not taking the lead of the sheep goat program. I started new program a year ago. We have now already more than 30 scientists working for me. We have, I think, the world's strongest team on this small ruminant. We should collaborate to work on the the Indonesia breed. Thank you very much.
Thank you very much, Prof. Han.
All right, thank you, Pak Dr. Ihsan.
Thank you for, Rama, for your moderate nicely. Thank you.
All right. Okay. Now we have to close the session. Well, on behalf of the Dean of Animal Science Faculty, Hasanuddin University, we would like to thank all the speakers, especially Professor Jianlin, for the great talk and information from your presentation. And of course, to the audience for your time to join this event. And thank you for your attention and have a great day. Thank you and see you in the next event.
Yes, thank you. Thank you. Have a good day. Bye-bye.
Bye.
Thank you, Prof.
Yes, thank you, Isa.